So, hello and good afternoon. There will be one presenting, and then my colleague Line will be online and help you in the chat. But I'll just share my screen and get started. So today I will be talking about how to use microsensors in biogeochemistry and microbial ecology. Later this afternoon, my colleague Line will talk about the use of microsensors in microbiology, biomedical sciences, and renewable energy. Okay, so just a few words about myself. I'm application scientist here at Unisense. I've been that for a bit over 5 years. Before that, I worked for many years as a scientist at the National Environmental Research Institute and Aarhus University here in Denmark. So I've done research mainly on marine nitrogen and carbon cycling, and microsensors have been part of my toolbox in that work. So the short outline of my presentation today, I will discuss a little bit about calibrations and temperature compensation, which is important for doing measurements and getting them correct. Then I have a section about monitoring concentrations over time. I'll show you the microrespiration system and the stock sensor and some data obtained with that. Then I'll go into the microprofiling part, show you something about what to consider when to set up the experimental design for microprofiles, and also something about sediments and microbial mats data from these studies. And finally, I'll discuss the laboratory versus field measurements, because we also have some field systems that allows you to take the sensors out into the field. So to get started with the calibration. So first of all, what you want to do is when you do a calibration, that is you want to establish a relationship between a sensor signal and a concentration. And this is what you do in a calibration. So the simple way to do this is to always calibrate and measure at the same temperature and the same salinity, because then you don't need to consider anything, any compensations. All our gas sensors for oxygen, hydrogen, nitrous oxide, nitric oxide, and sulfide are linear. So we generally recommend a 2-point calibration. So 1 0 and 1 known concentration. And then the sensor is calibrated. We have calibration kits to help you do these calibrations. We have them for sulfide, nitrous oxide, oxygen, redox, and pH. They come in a box like this with equipment for 10 calibrations. They come with syringes and a calibration cap and can snap onto the protection tube of the sensor. So you can inject your calibration liquid here. And then it comes with whatever you need for mixing and stuff. And it's a bit different for the different calibration kits. It's a very handy thing, at least to take to the field, for example, because then you don't need to bring a lot of chemicals. All this can be shipped very easily. Without any dangerous goods declarations. So I'll first discuss the oxygen calibration and go a little bit into detail with that, because that's similar to some of the other gas calibrations. So generally, as I said, we do a 2-point calibration. So for oxygen, that will be air saturated water typically and an anoxic solution. The oxygen concentration in air saturated water, that's well known from the literature. There are numbers of tables with the concentration as a function of temperature and salinity, and that's all built into our software. So you can actually get the concentration of oxygen once you have an air saturated volume of water and you know that it's temperature and salinity. You just enter that into the box here, the pressure, the atmospheric pressure where you're working, and then the content of the gas in your calibration gas. So in air, that will be 20.946% oxygen. That's written in ppm here, and then you get the concentration in micromoles per liter. This can be done for oxygen, hydrogen, nitrous oxide, and sulfide gas. So first, it's important to realize, as Lars also mentioned many times, I'm sure, is that the sensors respond to the partial pressure of the gas and not to the concentration as such. And this has implications for how to calibrate. Henry's law says that the concentration in water equals the solubility of the gas times the partial pressure of this gas. You can rearrange that, and so the partial pressure equals the concentration divided by solubility. And if you keep the solubility constant, then you will get a change in partial pressure, will give a change in concentration. And this is actually the reason that we can measure concentration with a sensor that measures partial pressure, as long as we keep the solubility constant, and that means keeping temperature and salinity constant, and that's normally not a problem. So salinity affects the gas sensors, the calibration of the gas sensors. I will discuss two situations. Situation one is if you are bubbling with a gas, could be you're bubbling with air through water to create air-saturated water. What you're actually doing is you are getting the same partial pressure of oxygen in the water as there is in the air around it, or the air you are bubbling with. If you then add salt to this, then you will change the solubility, of course. But because you are continuously bubbling with air, you will maintain the same partial pressure. And that means that a sensor signal will not be affected by adding salt here. So you will have the same sensor signal at different salinities. So in this situation, the sensor signal is not affected by salinity. And that's only because you are maintaining or constantly maintaining the same partial pressure, and the sensor responds to partial pressure. The other situation you could have is that if you have a stock solution and you want to dilute that into a concentration or calibration, this could be, in this case, there's a nitrous oxide saturated water in this bottle, and we dilute some of that into a volume of water to get a concentration. Now, if we do that at different salinities, you'll get a totally different result than the previous. Here you'll see that the sensor signal will be different at different salinities. So it will be higher at higher salinities. Now, the reason for this is that the partial pressure of the gas is affected by the salinity, and the concentration, the solubility changes with salinity. So when you add salt, you decrease the solubility of your gas, and then the partial pressure has to increase because you have the same concentration. So that's why you have higher sensor signal here, because the sensor measures partial pressure. And this is because of Henry's law, that if you change the solubility, in this case, it's being lowered, then and the concentration is kept constant, then the partial pressure will increase and the sensor signal will increase, as you see here. This sulfide is a special case also for calibration, because the sensors measure the H2S form of the sulfide, whereas this total sulfide pool consists of the H2S gas, that's dissolved in water, plus the HS minus and the S2 minus ions. They're in a pH-dependent equilibrium, as you can see here. So at neutral pH, you have about equal amounts of the H2S and the HS minus. We recommend to calibrate at pH levels below 4, so you have almost all of your sulfide in the H2S form, which is what the sensor is measuring. The calibration kit actually has a pH of 2.1, so it's down here, so you have 100% of the sulfide in the H2S form, so you know the concentration of H2S in your calibration liquid. Again, we do a 2-point calibration, and what's also important here is that if you go up at very high pH, above 10, for example, you see the H2S is very low, so the fraction of the total sulfide pool that's in the H2S form is extremely low, and the sensor will only measure this. So in reality, it will not measure anything. So you cannot use the sensors above pH 9 or 10, at least not at normal concentrations of sulfide. The sulfide calibration kit is very handy because it comes with with sulfide precipitated as zinc sulfide, and then some hydrochloric acid, 10 vials of the first, 10 vials of the second, and all the accessories. And then the idea is that you shake this suspension of zinc sulfide very well, and then transfer 1 milliliter to the hydrochloric acid vial, and you'll then within 3 minutes have all the H2S in the all the sulfide in the H2S form, and you can do the calibration. When we do a batch of these zinc sulfide vials, we measure them against the certified H2S gas, so we know the exact concentration that will be in the vial once you've done the dilution here. And this is written on the front of the box, so you'll know the exact concentration here. Don't worry about anything with crystal water or anything, because it's calibrated. So there are other things affecting the sensor response. One of the important ones is temperature. As you see here, for an oxygen sensor, the sensor signal increases with temperature for the air saturated water, but also a little bit for the anoxic water. So the zero signal for the sensor also increases. So there are different things to consider here. First, in the Uniamp lab instruments, we have a temperature compensation for the gas sensors within ±3 degrees from the calibration temperature. But in order to do that, when you calculate a concentration from a sensor signal, so in reality you use the calibration to get back to a concentration from a signal. This is divided into two steps now. So first we convert the sensor signal into the partial pressure. Partial pressure is then converted into concentration. And we do that because the temperature affects both of these parts here, 1 and 2, but it affects it differently. The first one we call the sensor response, the second one we call the solubility response. So when you convert a signal to concentration, what you would like to do is simply just have a millivolt signal and convert that to a micromolar signal. But we split that into first converting the sensor signal into a millivolt signal, the millivolt signal into a partial pressure signal here, and then that is converted into a concentration over here. Sensor response and solubility effect, and both of these are affected by temperature, as I mentioned, and differently. You can, in the software, decide if you want to use this or not. There's a solubility temperature compensation here and a sensor temperature compensation here. You can choose one or the other. If you use the sensor temperature compensation, or if you're working in gas samples, for example, you will not want to do a correction for the solubility. You just want to do a correction for the temperature response of the sensor itself. Okay, a few recommendations about calibration. So always calibrate and measure at the same temperature if you can. The Uniamp instruments, lab ones, they give you the option of ±3 degrees centigrade temperature compensation. So ±3 degrees from the calibration temperature. Be careful with the baseline, so that the zero calibration. This is especially important at low concentrations. For example, if you're measuring sulfide, you want to know if there's a little bit or there's nothing. Then you need to know the exact signal for zero to be able to tell the difference. Always calibrate your sensor before doing the measurements. First of all, you need to know if the sensor is working as you expect it to. And second, if you manage to break the sensor halfway during the experiment, you'll still have data until you break it. And calibrate often. That will give you a better precision. Okay, so I'll get into the part about monitoring concentration. First about the microrespiration system, and then I'll show some data from the stock sensor. So the microrespiration system is a system designed to quantify metabolic activity of small organisms in a very small volume. The basic unit here is this glass vial, where you can have your organisms. It could be a microbial suspension, it could be a small organism, a crayfish, a fish larvae, or whatever. And you have magnetic steering inside, and this is placed in a rack where there is place for up to 8 of these small vials, with a magnetic steering underneath each of the vials, and you can control that from the stereo controller here. The sensors are mounted in this blue aluminum guide so that it can be placed safely inside the sensor. So the chamber itself, or the vial, has a glass lid with a glass seal to the vial. There's a small capillary opening in the lid here, 0.7 millimeters in diameter and 15 millimeters long. And this narrow capillary is so narrow that there's no convection, so there's no transport of water through this channel. There can, of course, be diffusion through the channel, but because of the 15 millimeters length and the very small cross section of this channel, the exchange of solutes through this is so low that you cannot see it. For example, if you have an anoxic volume of water here and leave it over a day or two, you can still not see any oxygen in there, even though there's air saturation outside. The capillary here allows for insertion of the sensor. The sensor must be placed in the rack here so that it's aligned above the vial. And when you then twist and push the sensor in through this aluminum guide, the sensor tip will be inside the sensor and you can do the measurements. The vials here, the microrespiration chambers, they come in different sizes, from 400 microliters up to 4 milliliters. If you want 8 of them in the rack, there's room for 2 if larger ones, medium-sized ones, and then we have the large ones as well, looking like this. There you need to create your own magnetic steering and a holder for the sensor, but we can get them made in any size from our glass blower. All our sensors can be used in this, so you can work with any of them. And there's a software that shows you, first of all, real-time data, but also collects data from the measurements and keep them organized for each chamber, so you can work with up to 8 chambers at the same time and still collect data and keep them in order. The way the software works is that you can measure, you can see the concentration change here, oxygen decrease during the measurements. In this case, there's been 2, chamber 1 and chamber 2 here. The oxygen concentration has been read several times, and there's breaks in between where the sensor has been used in the other chamber. As whenever you're doing measurements, you will immediately see the slope, so micromoles per liter per hour. A change here, you'll see an R² value and the standard deviation, so you can see the quality of your data and see for how long you need to measure to get a good data. The software also calculates the rates of change. We have 2 ways of doing that. So there's one called rates within, where you see there's a linear regression, the yellow line on top of the green data points within each period where the measurements have been made. And that gives a series of data you can see down here. We can also use the rates between, and then the software calculates the change during the break when there has not been any measurements. So it takes the last points before the break and the first points after the break, and then makes a calculation of the change from before and after this break here. And it does that for all of these. You can select how many of the data points before and after the break you want to include, and you get the data here. You can get all sorts of statistics else as well. And I just want to mention also that we have the double chambers like this. So in this case, you can have 2 sensors sitting in the same volume of water, so you can measure simultaneously with 2 sensors. And we can also do that for 3 chambers or triple chambers. You can have 3 sensors at the same time if you want. An example of what it can be used for, here is a small jellyfish, 3 to 5 millimeters in diameter. It was placed inside a 2 milliliter chamber. They used this metal stainless steel mesh, which is placed on top of a glass ring, and there's the glass coated steer bar underneath, so that the volume here is mixed, but the organism is not crushed, so it's kept protected. And then they measured oxygen concentration during the experiment, and they did that in summer and winter and compared different feeding rates and so on, and saw how that would affect the respiration rate. So we also have a sensor for low oxygen concentration. This is called the stock sensor, switchable trace oxygen sensor. That can be used both in a lab and the field, and I'll show you some data from that. It is for trace amounts of oxygen and typically has a detection limit of 10 nanomolar of oxygen. So very, very low oxygen concentrations. The way it works is that it has it's actually 2 sensors in one. The inner sensor here, the red glass walls here, is sitting inside another sensor. This is the measuring sensor. So that's what gives us a signal for oxygen. This sits inside another sensor, which has a large porous gold cathode here. And when this one is switched on, so this is polarized, then oxygen that enters the silicone rubber membrane here in the front is being consumed on the gold cathode, and nothing gets into the measuring sensor. So the measuring sensor will then show the signal for 0 oxygen. When we then switch the front sensor off, or the front guard as we call it, then oxygen will pass the silicone rubber membrane past the porous gold lump here and the silicone rubber membrane here, and gets measured inside the measuring sensor. Then we'll get a signal for oxygen. So we can switch back and forth between the signal for 0 and the signal for oxygen. The way it looks is like this. So the blue one shows this signal from the measuring sensor. So this is 0 down here, and the orange line is 100. That means it's the front guard is switched on, so nothing, no oxygen comes into the measuring sensor. When we then switch the front guard off, then we get a signal for oxygen. Switch front guard on, we get a signal for 0, and for oxygen, 0, for oxygen, so on again. What we like to do here is that we want to measure the difference in signal between the front guard off and the front guard on, which we call the delta signal. In this case, I did an experiment. I had a little bit of oxygen in the bottle. I added 1.37 micromolar oxygen twice, and you see this, the difference, the delta signal increased with the additions. Looking more in detail, this is when oxygen is present. You can calculate the difference between the signal from the sensor with the front guard off and with the front guard on. So that's the dark periods down here on the x-axis. If there's no oxygen present, then the delta signal is simply 0 because there's no difference. And this means that when the delta signal is 0, this gives a very simple calibration, because when we have 0 oxygen, we have a 0 signal. So the delta signal is 0. And that means that the calibration curve will always go through 0.0 here. And we can actually get away with a 1-point calibration, which can be handy now and then. And the concentration is simply calculated as the delta signal divided by the slope. So very simple calculation. I'll show you some data from some cruises I participated in way back, a long time ago, along the coast of Chile and Peru in the eastern southern tropical Pacific. It's known for its oxygen minimum zone. This is shown here in red. So this is a big cloud of oxygen or low oxygen waters floating around just outside the coast of South America and stretching out into the Pacific. It starts at about 100 meters of depth and is about 200 to 250 meters thick, and it has a long extension. It was not really known what the exact concentration was in this until we got the stock sensors into there. It was known that it was low, probably below 2 milligrams per liter, probably below, in some cases, 2 micromolar per liter, depending on the instruments used and the detection limits of those. But what we had the option to do was that we had stock sensors with a detection limit of 5 to 9 nanomolar. This was in the early days of the stock sensor. And we have a custom-built amplifier that was mounted on a CTD, so we could do profiling in a lot of different stations along the coast here. This shows the cruise track, and this shows the data. So I'll show some data from an area close to shore here. This is the one shown here. You can see there's oxygen in the upper about 100 meters, and when we get below that, the data points cluster around the 0 here. So all these data points were not different from 0, so oxygen was not detectable by the 5 to 9 nanomolar oxygen detection limit. We had different stock sensors, so they were a little bit different in detection limit, but generally there was less than 5 to 9 nanomolar oxygen here. When we got further offshore, that station here, there was always a little bit of oxygen present, 50 nanomolar or so oxygen. It was very consistent. So apparently there was slightly oxygenated out here, but very low in oxygen in here. We did measurements all the way along the coast here, and there was a stretch of about 1,600 kilometers where there was that looked like this. So with oxygen below the detection limit of these 5 to 9 nanomolar, about 200, 250 meters thick and about 100 to 150 to 175 kilometers wide zone. So a huge volume of water. So you could ask, does it really matter if there is 2 micromolar, there's 10 nanomolar of oxygen? Well, it actually does. And we did some lab experiments to investigate that. We had reactors where we could mount the stock sensors in. These were glass reactors with a PVC piston, but coated with glass, so as much glass as possible to avoid contamination with oxygen in the water. With the piston here, we could take samples without introducing a headspace, and that was important because we were doing 15N experiments that we wanted to collect N2 and measure the isotopic composition of that. We could manipulate and measure oxygen in these, the type of data we got, like this. So this is an experiment where we added 15 in nitrite. We were interested in measuring NMOX and denitrification, and they will, from isotope pairing, they will form 29 or 30 N2 gas. We measured that on mass spectrometry on discrete samples. You can see 29 and 30 N2 here. But we were able to control and measure the oxygen. That's the blue line here. After filling the reactor with water, there was always some contamination. So we had to bubble with helium to get the oxygen out. We got down to a couple of nanomolar. Then there was a little bit of leak into the reactor. So we got up to, in this case, about 15 nanomolar, and then a little lower at the end of the experiment. But we were able to know the exact concentration of oxygen. And we did a lot of manipulation experiments where we added more and more oxygen to see how that affected the production of N2 and these NMOX and denitrification processes. And we were interested in seeing how much oxygen was needed to inhibit the processes. And we calculated the C50, which is the amount of oxygen required to inhibit the process 50%. For N2 production by denitrification, it's shown here in the inhibition as a function of oxygen concentration up to 2000 nanomolar. And already at about 200 nanomolar here, we had a 50% inhibition of N2 production. So that we calculated the C50 to be 205 nanomolar oxygen. The nitrous oxide production by denitrification was less affected by oxygen. Data was not nearly as nice though, but about 300 nanomolar of oxygen. And N2 production by NMOX, not so nice data, but the FIT gave us about 900 nanomolar of oxygen required to inhibit the process 50%. We extended these experiments to other processes. So these were the ones that I just showed you here, the 50% inhibition concentrations. Nitrite reduction to nitrate, also a dissimilatory process, was inhibited 50% by about 740 nanomolar. The ammonium oxidation to nitrite and the nitrite oxidation to nitrate was, we also looked at that, and that where we measured the Km value, and there was about 330 and about 800 nanomolar of oxygen. So the important thing here is that all the regulation of the important processes in the nitrogen cycle that takes place in the nanomolar range. So it's important to actually be able to measure in that range. To do so in situ, you can work with the field data logger mini here. It comes in a 2 and a 4 channel version. It can go to 6 kilometers water depth. The sensors are mounted on the in situ amplifier here and are pressure compensated so they can stand the pressure. It has a battery for up to about 20 days of operation, and you can interface it with a CTD or other equipment if you want. You can also use stock sensors in the lab. Now it's much easier than used to be because the Uniamp instruments, they have now built-in stock support. And that means that when you plug a lab stock sensor into here, it's only one connector. Then you get access to this control panel where you can set the front guard on and off times in seconds here. You can set polarization and so on. But the important thing is it's very easy, plug and play. You can plug it in and it's running. The type of data you get were a bit annoying to analyze until we got it, an analyzer built into the software. This is the type of data you get. The blue line shows the sort of the rhythmic signal from the measuring sensor with the front guard off. It shows a high signal with the front guard off. It goes down to the 0, so up and down all the time. The reddish line down here shows the front guard signal, whether it's on or off. But with the analyzer that's now in the software, you can easily analyze it. Here you see a couple of these cyclic patterns. See the red is the measuring oxygen sensor, goes up and it goes down again. What I've done here is I have, with the mouse, you can push these dotted lines, so select which data points you want to include. I've chosen the 5 last points here before the front guard is switched on, so for the measuring signal, and the last 5 points of the oxygen signal before the front guard is switched off. And what you get is for each of these, then you just click the Analyze button and it runs through the whole data set. You get a time for the measurement, you get a value for 0, so that's the average of this point, and you get a value for measurement, that's the average of these 5 points, and then it calculates the delta signal as well, and you can export that to Excel or CSV files. So very easy to use now. Okay, so I'll go to the other part, talk about microprofiles. So discuss the microprofiling system, a little bit about a theory about microprofiles and diffusion, something about experimental design, and some data examples. And then I will go into showing how the photosynthesis can be measured in detail with the light dark shift technique. So the microprofiling system looks like this. It consists of a micromanipulator. It can be motorized or manual. There's this heavy lab stand, so you can have a vibration-free mounting of the sensors. There's the software that allows you to operate the motor here and collect the data. There's an amplifier, in this case an FX6 uniamp, where the sensor is connected to, and there's a motor controller that will operate the motor. And then you can get the nice data you see over here collected automatically if you have the motor control. Such a microprofile has a lot of information in it. So this is an example from a biofilm with an oxygen production in the upper layer. This is shown by the green bar here, which this is consumption rate. So a negative consumption rate is the production rate, and then consumption down in the lower part. So the qualitative information you can get from profile. First of all, if there's a straight line here and there's only diffusion going on, this is the case in the diffusive boundary layer, which is a thin layer of water of a few hundred micrometer thickness that due to friction between the surface of the biofilm here and the water is more or less stagnant. At least the water movement is so low that convective transport does not play a role. It's only diffusive transport. And when that is the case, you will get, and there's no consumption and no production, then you'll get a straight line, as you see here. Below that, we have a curvature that goes this way. And that indicates there's production of oxygen in this case. And finally, below, we have a curvature going that way, and that indicates consumption of oxygen. Again, this is shown in these bar graphs here. A little bit more into detail. So this is a one-dimensional system. I've shown an oxygen profile here. We have well-mixed water phase here. This part here is the diffusive boundary layer, and then we have a sediment or biofilm consuming oxygen in here. So the quantitative information you could get from this is, well, first of all, you can of course see the concentration. So if you're interested in knowing how much oxygen is available for microorganisms in your biofilm, you can read that directly from the graph. If you have other profiles, pH, sulfide, and so on, you can see if there's overlap zones and so on, and get that kind of information. You can also do flux calculations. So you can calculate the flux of oxygen, in this case through the diffusive boundary layer, by finding the slope of the profile here, multiplying it with the diffusion coefficient of oxygen in water. That's called J water. That's the flux into the sediment. You can also do flux calculations in the sediment, JZ here. It's a little bit different. You still need the slope, dCdx here, but then you will need to multiply with the porosity of the sediment or the biofilm, and then the diffusivity of oxygen in this sediment or biofilm. And the diffusivity shown here can be calculated different ways. The simple form is just to multiply the diffusion coefficient of oxygen in water with the porosity. We can also do that in different ways, and I'll get back to that later and in the presentation tomorrow of how to analyze the profiles. And finally, you can also analyze the curvature of the profile here. And that will give you the, in this case, consumption of oxygen shown by this bar graph here. And what you need to do is you need to take the second derivative of your concentration profile, multiply with the diffusivity and porosity again. Then you get the net consumption. And this can all be done in the Unisense software, the profile part. We have written a small application note about this, 6 pages one, where you can see how to calculate oxygen consumption rates using the software. You can download this from our website if you want. Just go to Knowledge, Application Notes, Biogeochemistry, and you'll find the application note here, profiling application. I will go through this tomorrow at 4:30, show how to use the software for this. So when you do a setup, want to make a setup for measuring microprofiles, there are things to consider. First, we would want a steered and well-mixed water phase. So here I show two situations. Here's a not well-mixed water phase. If you don't have any active mixing of the water phase, you will have a sluggish mixing. So there will be a bit of air movement that affect the water. So there will be a little bit of circulation. The evaporation on the surface will cool it a little bit. They'll sink in and create a little bit of circulation here. And the oxygen will be consumed on the sediment surface here. So you will get heterogeneous water column with different concentration of oxygen in the water column and also varying concentration of oxygen on the sediment surface. And that will not lead to a steady state. If, on the other hand, you mix your water phase well and keep the oxygen constant up here, then you will have a constant concentration on the sediment surface and you will have a steady state profile over time. So it means it will not change over time. This is important for doing all the calculations that I just mentioned. An example is here where there's sediment cores in a bucket, thermostated bucket with cooling. There's a microsensor coming into the sediment here, and there's a bubbling in the bucket that creates circulation, and there is a plastic pipette where air is blowing across the water surface that keeps so it creates a flow, a one-directional flow across the sediment surface. That's a good way to get a steady state and a well-mixed water column. Then you also need to know where you're measuring, even if you're in the sediment or not in the sediment. One way to go is, of course, a visual observation through a dissection microscope like this. This is actually a setup we sell that is excellent for working here. It has a long focal distance and a very sharp picture. It has a camera, and you can collect data pictures actually in the Unisense software using this size microscope. You can also use a USB camera to get pictures of what you want to observe your sensitive relative to whatever you are measuring. Another way of doing it is that if you have two sensors, for example, oxygen and sulfide, like in this situation, we have a two sensor holder for the micromanipulator, where the second sensor sitting over here can be manipulated relative to the first sensor, so you can get the tips of the two sensors very, very close together. There are a number of fine controls here on the micromanipulator that allow you to tilt and twist and move the sensor, second sensor relative to the first. And then you can get the tips very close together. And this is a big advantage for oxygen and sulfide, for example. This is an oxygen profile, and the sensor for sulfide was placed with the tip very close to that of the oxygen sensor. It's relatively easy from an oxygen profile to guess where is the sediment surface. So it should be around here, where the oxygen has started to decrease and is still linear decrease before it starts to curve. So a good guess would be the surface of the sediment is here or maybe up here. But because we know that this would be very difficult if you only had this profile here, because you'd see sulfide at some point at depth, but you don't know where it is relative to the sediment surface. But when it's mounted together with the oxygen sensor and well aligned, you can interpret these, and you can, for example, see that, okay, the bottom of the oxygen zone is here, the top of the sulfide zone is here. So they reach each other, but they do not overlap in this case. So that's the double sensor holder is really nice for things like this. Second thing you want to have control of your water condition or incubation conditions, you could say. So temperature, oxygen, and nutrients are important, so you get to what you measure in the lab also reflect what you see in the field. Here's a group of scientists in Iceland. There's a hot spring coming out here. Water is flowing down over this area, creating these nice laminated cyanobacterial mats. Water temperature is 60 degrees, so it's really hot. There's no macro life. That's why we get these nice laminated systems. These guys took the samples inside in a flow channel where they had water from the site, heated it to 60 degrees and created a flow across the mat. And then they could measure with these sensors oxygen concentration in light and in darkness. And so that's a really good example of how to recreate in situ conditions in the lab. A better way, you could say, would be to go out into the field. This is from Yellowstone. Michael Kühl is lying here in the good old days, measuring with a hand-operated micromanipulator and a technician taking notes. Now we have the field profiling system where there's the field multimeter and the field motor here with the micromanipulator on, and you can operate everything from here and collect data and so on. Very easy to do. The field multimeter is a splash-proof meter. It can be operated either, as you saw in the picture, independent of a PC, directly from the keypad that's on the instrument. You can also connect it with a cable to a PC, up to 70 meters long actually, and operate it just like a lab system. The field motor here is waterproof, down to 10 meters, and you can place it wherever you want, and mount the sensors either in this sensor holder here or put a micromanipulator on, and you can do measurements. What they did here in Yellowstone was they were looking at hydrogen production in these microbial mats. They were working diurnally, so many profiles over night and day. Here's one in light from afternoon, high sunshine. Have a high production of oxygen. See the blue one here, a large peak of oxygen that creates this high pH value. Sulfide is pushed down below the oxygen zone and hydrogen is not present. Then right after sunset, in darkness, then a peak of hydrogen suddenly appears up to 22 micromolar of hydrogen. Oxygen has now decreased down to the to a low level compared to the light situation, and the sulfide profile moves up and follows on the bottom of the oxygen profile. This is a type of measurements that's easy to do when you're out there because you have in situ temperature and light conditions and so on, water running across the surface of the biofilm and all that. It would be more difficult to recreate in the lab. So as I mentioned, photosynthesis can also be measured in detail using the light dark shift technique. So the principle here is that you have a microbial mat. It has to be a dense microbial community like this. And anywhere in this sediment or microbial mat, oxygen concentration will be constant over time if you have steady state. And when the concentration is constant, that means that all the produced oxygen by photosynthesis, that must be removed. And that's removed by respiration and by diffusion away from the site. If you then suddenly turn light off, then the oxygen removing processes are not changed. This was the respiration and the diffusion, and they are driven only by the concentration, and the concentration has not changed. However, the oxygen production is stopped. And that means that the oxygen concentration will now start to decrease with a rate that's equal to the production of oxygen in light. So the missing production of oxygen is now shown as a decrease in oxygen concentration. What you need to do to do that is the light sensor, so you know exactly when the light is switched off, and you need a very fast responding oxygen sensor, 90% response time of 300 to 400 milliseconds. We have those, no problem. If you have a microbial mat here, a very active photosynthetic microbial mat in darkness, you see it, and place the sensor in the surface layer, you can see there is constant oxygen concentration. And then switch on light, oxygen increases until it reaches a constant concentration. Then we are now at steady state. Light is turned off and it starts to decrease immediately after turn off of lights. If you look in detail, and this is a screenshot from software, you have a steady concentration of oxygen until light is switched off, and then the oxygen concentration starts to decrease. The signal from the light sensor makes it possible to get the zero for the time axis here. So we know exactly when this break occurs. Software then calculates the slope of the decrease in oxygen concentration here, and that is then equal to the production of oxygen that was just before we turned off the light. The type of data you get are like this. So this is the green bars show the gross oxygen photosynthesis measured with this light dark shift. So these are shown in bars here for every 200 micrometers into the biofilm here, or the sediment. So this is what you get, the directly measured gross oxygen production. But you can also do the flux calculations here. So you can calculate the flux upwards and the flux downwards from the zone where oxygen is produced. And from that, from summing these two up, you get the net oxygen production. And then you can actually also calculate the respiration in light simply by subtracting the gross oxygen, the net oxygen production from the gross oxygen production. So it's possible to measure respiration in light, which is not possible in any other way. You would typically have to incubate the system in darkness to see the respiration, but in this case, you can actually do it in light and at the relevant oxygen concentrations. If you did this in darkness, oxygen concentration would be very low, and oxygen penetration would also be much lower. So I already touched upon this, but it's now and then a big advantage to go into the field to do measurements. You get to avoid some problems that you can create for yourself in the lab. Here's some examples of where you can take the systems out. This is the very simple way to do it. So it's simply just a micro manipulator and some sensors, and you can go out and measure in shallow waters. This is the more advanced, where we have a free-falling lander that's dropped from a ship. It sinks to the seafloor and does its measurements and comes up again when you ask it to. We have small application notes discussing sort of the advantages of lab versus field, and this is Ole Pedersen from Copenhagen University. His mantra is learned in the lab, explore and confirm in the field, and this is explained in this winning formula application note, which you can also download from our website. So I'll show some examples of where it's an advantage to go to the field instead of just going and doing work in the lab. So here's an example of some sediments from the coast of Chile. An example of 3,000 meters water depth where there's an oxygen profile from the lab and measured in situ. And so that's quite a difference in oxygen penetration. And they did this more systematically by comparing this difference and aligning it with water depth. You see the deeper waters, the bigger a mistake you make. So when you go to 5 kilometers water depth, you actually have about a factor of 5 difference in oxygen penetration between lab and in situ. And then the calculated diffusive oxygen uptake makes more or less the same mistake, so to say, that you get a far too high oxygen uptake calculated in the lab compared to what you can calculate from the field data. Yeah. Another example of data from a much shallower area that is from an intertidal flat in the English Channel on the French side. They did a 6-day experiment placing one of these mini profilers in the intertidal zone. They got a lot of these, actually 900 microprofiles of oxygen under different conditions. In the light, there was a production of oxygen. In the dark, there was a consumption of oxygen. And this is a type of experiment that would be difficult to do in the lab because they were having a lot of variation here. So this continuous line here, that shows the water level. The horizontal line shows the position of the sediment. So whenever this water level is higher than the sediment, you have inundated sediments, a water cover sediment. Those are the gray periods here. And when it's lower, the sediment is exposed to air. The temperature also varies a lot over time. That's the dotted data here. And down here, you can see the net oxygen production, and you can see the light level with this continuous line here. And the dotted oxygen, the net oxygen production was calculated from these profiles simply as fluxes. And you can see they can go from positive to negative in a very short time. So this gives you much more information and much more correct information than you can get in the lab. It will be very difficult to create this scenario in the lab. So we have different options for field systems. As I mentioned, we have the mini profiler where you can do microprofiling in situ. Here's a diver guiding it into place. You can also just lower it from a boat. We have a mini chamber lander where there is a polycarbonate chamber here that encloses 30 by 30 centimeter area of sediment, so you can measure the fluxes in and out of the sediment. You can measure with sensors, but you can also take water samples, up to 12 samples during an incubation. And then we have the eddy covariance, where the flux of oxygen past the tip of the sensors here is measured and calculated. I'll just mention that in a moment. We have also the free falling lander option here, where we can mount any of these 3 systems on this. So the idea is that you drop it from a ship. It has flotation rack on top, and it has weights in each corner, and so it has a negative buoyancy when you put it out, and it sinks to the seafloor, does its measurements, and then with an acoustic release, you can then release the weights. That will typically be scrap iron that will be left on the sediment, and then this will float back to the surface, and you can collect the data from it again. So either deployed by winch or autonomous. The shallow ones, these mini profilers and the landers, are difficult to handle deeper than 300 meters, but this autonomous one can go all the way to the deep sea, or to at least to 6 kilometers depth. So these methods are complementary. You could say, though, the microprofiles, that will give you the idea of oxygen, very detailed knowledge of oxygen at one point, and you can calculate the oxygen consumption at different levels in the sediment. You can calculate a flux rate of oxygen into the sediment at this exact point where you have been measuring. And these profilers come in 1, 2, or 3 dimensional ones. So you can do either one profile, you can do a row of profile, or grid of profiles. The benthic chamber here will, in this case, I've shown oxygen concentration inside the chamber after the lid has been closed on 6 different stations in the Baltic Sea. You see oxygen decreases, so oxygen consumption can be calculated from the oxygen data. And then the eddy covariance technique, that typically that is used for oxygen fluxes without touching anything. So it's really a non-invasive technique. The principle is that this is an acoustic Doppler current meter. It measures the flow rate and flow speed or direction, in a very small volume of water, right where the sensor tips are here. So, and the sensors, they measure the oxygen concentration. And if you know the flow rate of water up or down, and you know the concentration of oxygen, you can calculate the transport of oxygen past the sensor tip. Now, this is in the turbulent layer, so it occurs very fast. So we need to sample fast, and these data are sampled, both oxygen and flow rates are sampled 60 times per second. Get a lot of data, but then you get a very detailed flux of oxygen. What this instrument does is that it analyzes sort of what has happened upstream from where the EC system is placed. And there's a footprint illustrated by this reddish layer. And depending on the structure of the sediment and the flow speed and current regime and so on, you can get anything between 5 or maybe 100 square meter footprint. But that means you can actually measure oxygen fluxes over a seagrass bed or a mussel bed or whatever, and get very good data on that without affecting the data and without breaking sensors into mussel shells and so on. All these systems, they are operated by our field data logger. Won't spend much time of this. It's a very simple to use instrument. You can program it very easily. Sorry, there's someone came in with a lot of noise here. Thanks. Okay. Yes. So okay. So it's operated through the Sensor Trace Suite software. There's a very simple programming tool here, which allows you from these blocks to make quite advanced programs. Actually, there's also even simpler remote user interface. Comes with 4 or 8 channels. It has a battery for 20 hours. We can add an extra 40 hours as an external battery. All sensors for our in situ instruments are connected via the in situ amplifiers. We have a shallow and a deep version. Sensors are connected here via the in situ connector. This one is filled with oil inside, and the sensors have a channel into the interior of the sensor where the oil can pass through. And that means that pressure outside will be transmitted through the silicone rubber sleeve into the oil and into the inside of the microsensor, and will have the same pressure inside and outside the sensor, and therefore they can be taken all the way down to 6 kilometers water depth or 600 bar pressure. And with that, I've finished my presentation. This is an example we got from Bruno Bomblé. They had its profiler out for a long time. It was totally overgrown up here, but the tips still keep themselves very clean. And somehow it managed to penetrate a piece of wood, probably rotten, and without breaking, you can see the tip coming out on the other side. Thank you very much for your attention, and I will now stop the sharing, and if you have any questions, I will be happy to answer those. And so there's a question: are there interferences between microsensors? No, the microsensors do not interfere with each other. There are interferences for the individual microsensors, and if you go to our website and go to the page for each sensor, you can go to the technical specifications and you can see what interferences there are. We know, for example, that an oxygen sensor, if that is exposed to high concentration of sulfide, you will see a decrease in signal. We know that the hydrogen sensor is affected by sulfide, but we also have a sulfide insensitive hydrogen sensor. So we try to get around these interferences, but you can see them there. And if you're in any doubt, you can write us, and maybe we can have some information about interferences. We've tested a number of different ones over the years, of course, and our customers have as well. Yes, the glass chambers can be autoclaved, the glass chambers of the microrespiration system. They can also be, but the sensors cannot be autoclaved. So the sensors can be chemically sterilized by ethanol or other sterilizing liquids. But the glass chambers, they are just like any other glassware in the lab. They can easily be autoclaved. Then another question: if you are profiling in a more solid sample, an organ, for example, what would the best way to be to secure that the sample, so the probe, is not moving the sample itself as it moves through? Yeah, well, that's of course important. That if you want to measure in an organ or in a muscle tissue or whatever, you typically want to use a very small sensor, because the smaller the sensor is, the less the chance is that it pushes the tissue away, and the better the chance that it penetrates. If, for example, we have people working in mice, they want to penetrate the skin of a mouse to measure any tumor inside a mouse. What they typically do is they use a needle to pierce the skin and then insert the microsensor through this hole in the skin, because the skin will be too tough for the microsensor to go through. So that's an option if you have a tough organ or tough sample. If there's a shell on top of it, you need to pierce. Another question is about the H2S sensor. Can the working electrode be poisoned by sulfide? No, I don't think it can. We have two different types of sulfide sensors, as Lars mentioned yesterday. The normal, typical one is the sulf sensor, where sulfide is oxidized to sulfate inside the sensor. It is linear up to 300 micromolar, but we can always make it less sensitive so that you can measure much higher concentrations. We recently sold one to a guy who wanted to measure up to 150 millimolar of H2S, which is extremely high. The normal one is 300 micromolar, but it's just a matter of making the hole in the tip of the sensor smaller so that less of the sulfide gets into there. But poisoning, no, that's not a problem. So if we use oxygen sensor to measure oxygen in unsaturated soil, will the solubility of oxygen in soil water and air phases affect the sensor signal? Yeah, that is a really good question. So the question is no. The answer is no. The reason is that if you have the sensor measures partial pressure, and if you have air and water next to each other and you have the same partial pressure in air and water, you will get the same signal there. So what we typically recommend people working in soil is that, and unsaturated soil, so with air pockets in it, there is to work with partial pressure instead of concentration, because that's the only thing you can be sure of. That is that you're measuring a partial pressure. Then you can always later translate the partial pressure into concentration if you want, if you know that the sensor has been in air, or if you know that the sensor has been in water.
Hello, this is Alida. Thanks for
Hi.
the presentation.
You're welcome.
I have a question because Lars yesterday mentioned that we have to be careful about the stirring sensitivity. And
Yes.
today you mentioned that it is important to have a flow of air in the surface of the core. But I wonder if I saw that the thickness of the core that you show us is very just few centimeters, the overlying water above the sediment, but sometimes the overlying water over the sediment is about 15 or 20 centimeters. So I wonder if this thickness of the overlying water, well, the flow on the surface is enough to cover this 50 centimeters of the overlying water.
Yeah, I think even if you have 15 centimeters of water, you should still make sure that it is mixed, so you have a water movement, and also that you are keeping the oxygen concentration constant. And the steering sensitivity of the sensor, well, if you have a standard oxygen sensor, it's typically around 1%. Sometimes you cannot really see it, so it's really low, so it will not affect your measurements. It may give you a result that is 1% higher than it should be when the water is moving, and so a very small error by this.
Okay. Yes. Yeah. Thank you.
You're welcome. So thank you. The NO sensor and the MR system, you already answered that, Lida, that you say we can make an NO sensor down to 2 to 3 nanomolar. Detection limit of the NO sensors down to 2 to 3 nanomolar. So really, really sensitive sensor. Thank you very much for listening, and let us know if you have questions, if you want to discuss more, and if you want to have a private session with one of us, then you are always welcome to write us. Thanks a lot.